single day.
Hi students, today we can see the next lesson, second lesson, biological classification. As we have studied in the previous video about that first lesson, the living world, we have seen all the organisms and the features of the organisms, and now we are going to see about how can we classify the organisms according to the different scientists and different kingdoms. Okay, 2 kingdom, 3 kingdom, 4 kingdom, 5 kingdom and 6 kingdoms. So all these classifications will be discussed in this class. Let us see now. First we can see the biological classification. Generally, the general definition for the word the classification is a grouping of organisms based on their similarities and dissimilarities. Organisms are classified by using the different features such as their similarities and their differences. The same characters and different features found in the organisms, it can be classified into different groups. So grouping of these organisms based on their similarities and differences is called as the classification. We can call it as a biological classification. So let's see the importance of the biological classification. You can see some phylogeny is given here, the evolutionary tracks here, and turtles, lizards, snakes, crocodiles. Okay, this is what we can say that phylogenus and reptiles are given here. And let us see now. So why do we these classifying organisms? Organisms are classifying or grouping or classification is made. What is the purpose of this classification? Easily identifying the organisms. Okay, for easy identification of organisms. To makes the study of organisms easy and convenient, helps to establish the evolutionary relationship among various organisms. So the classification is mainly made to easy identification of organisms, to make the study of organisms easy and very convenient and very comfortable, and it helps to establish the evolutionary relationship among the various or different kinds of or different variety of organisms. Types of classification. So the classifications generally can be classified into 3 types. One is artificial system of a classification, second one is the natural, and the third one is the phylogeny, means that evolutionary. Let us see the first one, the artificial system of classification. 1 or 2 morphological characters for grouping the organisms is called artificial system of classification. And next you can see the natural classification or natural system of a classification. It was based on morphology, physiology, ontogeny, anatomy, cytological and biochemical and taxonomy. According to these things, organisms are classified, and this type of a classification we can call it as a natural system of a classification. Phylogenetic system of classification is on the basis of the evolutionary relationship among the organisms. Phylogenetic always indicating the evolutionary link. Okay, so this system is based on the evolutionary relationships of organisms. So the types of classification can be classified into 3 groups: artificial system of a classification, natural system of classification, and phylogenetic system of classification. Let us see about the Aristotle and Ali classification. We can start now to classifying the organisms according to the different scientists. So very first you can see the name of the scientist is the Aristotle, and he first made the classification, and he made the classification is in a symbol basis. Okay, Aristotle is a Greek philosopher, and he used symbol morphological characters to classify the organisms. He is not using any deep characters. He is not explaining anything very intensively about the characteristic features of organisms. Instead, he used just the morphological, means visible characters of the organisms to classify the organisms. So according to him, the Aristotle, these organisms can be classified into only two groups. One is called as the plants and another one is called as animals. So Aristotle classified the organisms into two, one is plants and animals, that we know very well. He is the Greek philosopher. Okay, and the next slide we can see two kingdom classification. Okay, first let's see about the two kingdom classification. Two kingdom classification means that what we can say, that is the plants and animals. So this is called Linnaeus. This is a scientist and who proposed this theory, two kingdom classification. According to him, the organisms again classified into two groups, kingdom Plantae and kingdom Animalia. So these two groups are classified by this Carl Linnaeus, and Aristotle classified organisms into two groups, plants and animals, and a kingdom. Two kingdom is first invented by this Carl Linnaeus, kingdom Plantae and kingdom Animalia. Organisms divided into two groups. And when they say about the two kingdom of classification, there are a lot of drawbacks and a lot of points are missing and characters are missing in this two kingdom of classification. Let's see what are the drawbacks of two kingdom classification. First, you can see this system did not distinguish between eukaryotes and prokaryotes. We know very well about the eukaryotes and prokaryotes. I will explain you. Eukaryote as organism a true nucleus, and which organisms, those who do not have the true nucleus, call it as a prokaryotes. Eukaryotes, prokaryotes are belonging to unicellular organisms or single cell organisms. So in two system of two kingdom classification, there was nothing mentioned about the eukaryote and prokaryotes. Prokaryotic bacteria and cyanobacteria were placed under the plant kingdom. So this prokaryotic bacteria, so these are the unicellular organisms I told. So prokaryotic means organisms, they have the true nucleus, the well established or well developed nuclear membrane present, so that we can say the true nucleus present in the prokaryotes. Prokaryotic bacteria and cyanobacteria, these are all what we can say, it's grouping bacteria, but he kept this one according to the two kingdom of a classification. These two group of bacteria are kept under plant kingdom. That is also the drawback. And unicellular and multicellular organisms are not specified clearly here. Everything is kept in one kingdom, unicellular organisms and multicellular organisms. Protozoans were placed under kingdom Animalia. Protozoa, we have to study about the protozoans. So protozoans actually not Animalia, it is a Protista kingdom separately. And protozoans were kept under the kingdom, the Animalia. That is also one of the drawback of this two kingdom classification. And photosynthetic means the green algae, and non-photosynthetic, that is the fungi, right? So these organisms actually not mentioned specifically in this two kingdom, and they kept in one group, that is the plant kingdom. So fungi were placed under the plant kingdom. See that last point, fungi also kept under the plant kingdom. So this is also one of the drawback of the two kingdom classification. So let us see now the three kingdom classification. So three kingdom classifications invented by the Ernest Haeckel, the scientist name, in the year 1866. And according to him, you see that organisms can be classified into three groups. The first group is the kingdom Protista, and the second group is the second one kingdom is the kingdom Plantae, and the last one is the kingdom Animalia. So three kingdom classification invented by the Ernest Haeckel in the year 1866. According to him, organisms classified into three kingdom. One is the kingdom Protista, and second one is the kingdom Plantae. And next, let's see about four kingdom classification. It was invented by Copeland, the scientist name, and in the year 1938. According to him, the organisms are classified into four kingdoms. What are the four kingdoms? The kingdom Monera, kingdom Protista, kingdom Plantae, and kingdom Animalia. So along with the three kingdom, the scientist Copeland added only one kingdom, the kingdom Monera. So the four kingdom classifications means what are the four kingdom of classification? Kingdom Monera, kingdom Plante, kingdom Protista, and kingdom Animale. So this is what the four kingdom classification. Let us see about next the five kingdom classification. Five kingdom classification is invented by H. Whittaker, Robert H. Whittaker in the year 1920 to 1980. So according to him, the organisms are classified into five kingdoms. Okay, you can see here very clearly it is given. He included one fungi kingdom, then that four kingdom classification. You can see according to this Arch Whittaker that the organisms can be classified into five kingdoms. The first one is the Moneran, and second one is the Protista, and third one is the fungi, fourth one is Plante, and last one is Animalia. These Monerans and Protista kept under unicellular organisms, single cell organisms. You can see here Monerans are generally called as the prokaryote and the prokaryotes are called as the eukaryotes. I just explained about the differences between the prokaryotes and eukaryotes. So prokaryote means that absence of nucleus, or we can say the nuclear membrane are not established clearly, so the nucleus is absent. Protista means that the eukaryote, U means the true, karyo means it indicates nucleus. So eukaryotes means that true nucleus. And fungi, Plante, and Animalia kept under multicellular organisms. So this five kingdom is nowadays accepted one. Okay. So five kingdom classification: kingdom Monerans, Protista, fungi, Plante, Animalia. Monerans and Protista belongs to unicellular group of organisms. Fungi, Plante, Animalia belongs to multicellular eukaryotes. So now let us see about the sixth kingdom of classification. So this is the thing we are going to study detail in this lesson. So the sixth kingdom classification is first invented by Carl Oz, the scientist name. You can see the scientist here is given here. Okay. So according to him, the organisms classified into six kingdoms. Let's see about the six kingdoms. First, you can see the domain. From the domain, the kingdoms are derived. So the domain is classified into three groups. One is the archaea, and the second one is the bacteria, and the third one is the eukarya. Okay, so these are the three groups from this domain. So from this, the sixth kingdom we are going to see now. So you can see this archaea we can call it as archaebacteria, and the bacteria we can call it as a eubacteria, and this eukaryote, okay, can be classified into four groups. The first one is Protista, and second one is kingdom fungi, and third one is next one is the Plante kingdom, last one kingdom Animalia. You can see the six kingdoms are here. Now the domain is divided: archaebacteria, bacteria, and eukaryote. And archaebacteria classified into one archaebacteria. Bacteria means a eubacteria, the second kingdom, and the third one is the Protista. Fourth one is fungi kingdom, and the fifth one is Plante kingdom, and sixth one is Animalian kingdom. So you can see the order of classification is given here in the right hand side. The first you can see the domain. From the domain the kingdom is derived. From the kingdom the phylum, and from the phylum next to phylum class, then order, family, genus, species. So there are 8 cadres are here, the criteria. These are, we can call it as order of classification. First, the least one is the species, genus, family, order, class, phylum, kingdom, and domain. So let's say about the five kingdom of classification. Again, the Robert H. Whittaker, 1920 to 1980. We have seen that one. Okay, what are the five kingdoms? We have seen clearly the bacteria, Protista, Plantia, Animalia, and fungi. So this classification is mainly made on the basis of the mode of nutrition of an organism, the complexity of organisms means the body of an organism, then complexity of the cell, and then modes of reproduction of organisms. These five kingdom classification is mainly classified on the basis of mode of nutrition, complexity of organism, complexity of cell, and modes of reproduction. And last one, phylogenetic relationship. I have already told you about the phylogenetic relationship. It means that evolutionary relationship of organisms. You can see here the five kingdoms are given very clearly about that the different types of the mode of nutrition, complexity of organism, complexity of a cell, and a phylogenetic relationship. Let us see one by one here. The characters, you can see the cell type first. Mononerans are prokaryotic. Prokaryotic means absence of nucleus. Don't forget at any time. Protista eukaryotes, true nucleus present. Fungi also eukaryote, all eukaryotes. So nucleus are present in all other four kingdoms except mononerans. And second, you can see the cell wall, non-cellular polysaccharide plus amino acid. So non-cellular cell wall is present in mononerans. So cell wall is present in protista in some of the organism, and some of the protistants are not cell wall. Fungi present without cellulose, and plantae it is present cell wall and it is made up of cellulose, and animalia it is absent, cell wall is absent. In animals cell wall is absent. Okay, so polysaccharide plus amino acid is made up by the cell wall in mononerans, and protista in some cells present, and fungi you can see present without cellulose, but cellulose is present in plantae. Nuclear membrane. Okay, next. So mononerans no nucleus, so no nuclear membrane absent. Other four are eukaryote, nucleus present, so nuclear membrane is present. Fungi present, plantae present, animalia present. Body organization, mononerans all cellular, unicellular organisms, so we can say, and fungi onwards we can see multicellular, loose tissue found in the fungi. And then you can see in plantae tissue, organ system. Okay, and animalia you can see tissue, organ system, then organisms. Mode of nutrition. Mononerans are autotrophic. So here we can see chemosynthetic and photosynthetic and heterotrophic organisms, saprophyte and parasite. So autotrophs, we know very well, the organisms can make their own food by the process of photosynthesis, and it mainly required the chlorophyll pigments present in a chloroplast. So it's autotrophic, a group of organisms also present, and it can be classified into two types, autotrophic, chemosynthetic, and photosynthetic organism. Chemosynthetic using chemicals to produce the food, and photosynthetic we know using sunlight to producing the starch. And heterotrophic, some of the mononerans are depending other organisms for food. They are unable to produce their own food by the process photosynthesis. So saprophyte we know very well. Okay, that is a feed on the organism. Okay, next one, the protista. There are two groups we can say, autotrophs and heterotrophs. Photosynthetic autotrophs. Okay, protista. And fungi heterotrophic purely, and fungi are not having, they don't have the green pigments chlorophyll, so they will be unable to produce starch. So heterotrophs or the saprophytic organisms, we can know very well, it feed on the dead organism. Fungus, we can say the fungus, right? It feed on the dead organisms. And parasitic also, some of the fungi. And next to plantae, you see the mode of nutrition, autotrophy. Plantae, all the plants or the green plants, they make their own food by the process of photosynthesis using the chlorophyll, sunlight, okay, everything. Using inorganic substance such as the carbon dioxide and water, releasing oxygen. That's an important point. Along with that, producing the starch. Animalia is the heterotrophs, and they will not be able to produce their own food in the absence of the chlorophyll. Photosynthesis process found only in the green plants. And heterotroph, animalia or the heterotroph, there are two types of nutrition, holozoic and saprophytic nutrition. Let us see now one by one. Okay. So these are the general characteristic features of the five kingdoms: the kingdom Monera, Protista, Fungi, Plantae, and Animalia. We have seen some five types of the characters: the cell type, cell wall, nuclear membrane, body organization, and mode of nutrition. Let us see the next one. You can see here given in this picture very clearly about the kingdoms. So first two kingdom, three kingdom, 4, 5 and 6 kingdoms given. Key features of a kingdom. So you see the first to see the two kingdom. It is classified organisms into two kingdoms, right? What are the two kingdoms? Very first I have told you. It is actually given by Linnaeus in 1758, and the two kingdom is Plantae and Animalia. And three kingdom you see additional added Protista only, along with animal kingdom and plant kingdom. Protista added here, and lacked capability of a tissue differentiation, so that it is also failure. And three kingdom system is given by Ernst Haeckel in 1866. So what are the three kingdom here? Protista, Animalia, Plantae. So Plantae, Animalia, along with these two, Protista added here. Four kingdom classification. Again, along with these three, monerans are added. So studies showed that prokaryotes possesses a different nuclear structure. Okay, you can see it's given by the Copeland, scientist name in the year 1956. And what are the four kingdom? Monerans, Protista, Plantae, Animalia. And five kingdom, separate group of fungi. Here, along with that, the previous four kingdom, one kingdom is included, that is called as the fungi, and on the basis of five criteria. You can see it's given by Arch Whittaker. So what are the five kingdom? Monerans, Protista, fungi, Plantae, and Animalia. And six kingdom classification, three domains divided into six kingdom. So what are the three domains? Archaea one domain, bacteria one domain, and then the eukarya one domain. Archaea, archaebacteria, and bacteria, eubacteria, and a eukaryote domain can be classified. Fourth kingdom again, Protista, fungi, Plantae, Animalia. So the six kingdom totally we can say archaebacteria, eubacteria, Protista, fungi, Plantae, and Animalia. So you can learn this table very clearly, then you can come to know about very clear structure about the kingdom, different classification of a kingdom according to the different scientists. So see here, it's given here very clearly. Now we are going to see about that Whittaker classification, six kingdom classification, the Calvus. So first you can see the monerans can be classified into two groups. One is a eubacteria, another one is archaebacteria. Eubacteria, U means I already told you the meaning for the word the U. It means that the true bacteria. Archaebacteria, we will see one by one. So eubacteria is classified again into two groups. One is the heterotrophic bacteria, another one is autotrophic bacteria. This is according to the mode of nutrition. Heterotrophic bacteria, it is not able to produce own food by the process of photosynthesis. It has no the chlorophyll pigment, so that it is called as the heterotrophic. It depends on the organism for the food. Autotrophic bacteria can make their own food by the process of photosynthesis, autotrophs. This autotrophic bacteria again can be classified into two groups like hemoautotrophs and photoautotrophs. Hemoautotrophs, example sulfur bacteria, and photoautotrophs is the example cyanobacteria. Chemoautotrophs, it's chemicals, using the chemicals to make their own food. And autotroph bacteria, photoautotrophs, photo means light, and they are using the sunlight for the process of the photosynthesis. Again, come to this side, the archaebacteria can be classified into three groups. First one is the halophiles, second one is thermoacidophils, and third one is the methanogens. So the halophiles, generally we can see in salty areas, and archaebacteria you can see thermoacidophils. It looks like it can be survived in the hot temperature, hot springs. Methanogens is marshy areas, dumping areas. And next we can see kingdom Monera. We are going to see about the kingdom Monera detail. I told you in the previous slide, you can just check. Monoderans can be classified into two groups, Eubacteria, Archaebacteria. This classification we are going to see about monoderans only. Okay, so see here the kingdom Monera. So mostly the primitive form of life in the sense it is found during the time of very ancient, okay, very long, by very first it is a formed organism, so that we can call it as a most primitive form of life. And monoderans are prokaryotic and single cell organisms. Prokaryotic in the sense I told you, absence of nucleus, and then they all are the single cell organism. The organism is made up of only one cell, and that one cell performs all the life processes. And cell wall is made up of a peptidoglycan, except the Archaebacteria and Mycoplasma. Okay, so there are two groups are there, Archaebacteria and Mycoplasma. Except these two group of bacteria, all other bacteria in kingdom Monera, the cell wall is made up of peptidoglycan. And it is found everywhere, such as hot springs, deserts, snow, and a deep ocean. So these are the locations where the monoderans can be found very easily. So it's found such as a hot spring seasons, region of the hot spring, hottest area, and deserts we can see. Snow also it can present, and then the deep ocean also we can see these monoderans. Bacteria are grouped under the four categories based on their shape. So there are four different shapes are found in the bacteria according to their shape. There are the four shapes is a round shape and comma shape and spiral shape, like different shapes. Four shapes are there. So according to that, it can be classified into four types of a bacteria. According to the shape, the name is given. The first one you can see the cocci, and second one you can see the bacilli spore, and the third one is a spirilla, and last one is the vibrio. So these are the four different shapes and types of bacteria and monoderans. So this monoderans are classified into two groups. First I told you, one is a Eubacteria, another one is Archaebacteria. First let's see about that Archaebacteria. You can see here mostly primitive organisms. So monoderans are the primitive organisms. So bacteria, Eubacteria and Archaebacteria, both are the primitive organisms. So most primitive organisms, and they found in a harsh conditions. Okay, they are not found in a previous one like that, and they found in harsh conditions. And the peptidoglycan is absent in the cell wall. Instead, some proteins and non-cellulosic polysaccharide is present. I just told in the previous slide about that cell wall is made up of the peptidoglycan except cyanobacteria, right? Except Archaebacteria and Mycoplasma. So now Archaebacteria we are studying, peptidoglycan is absent in the cell wall. Instead of that peptidoglycan, some proteins and a non-cellulosic polysaccharide is present. That is a very important thing you must remember. Branched lipid chain is present in the membrane. So branched lipid chain is present in the membrane, cell membrane. And there are three groups of Archaebacteria we have seen. The halophiles is the first one, and the second one is the thermoacidophiles, and the last one is the methanogens. So halophiles are in the salty area, thermophiles are in the hot springs area, means hottest area. Thermo means the temperature, okay. And the methanogens found in the marshy areas, okay. Okay, let us see now. Next one. So next Eubacteria. So Archaebacteria classification finished, and next we are going to see about the Eubacteria is the true bacteria. So U is the word always mentioning. U means true. Don't forget at any time. So it is also called true bacteria. So this true Eubacteria we have classified two groups. One is photosynthetic autotrophs, another one is heterosynthetic autotroph. Okay, so photosynthetic autotrophs, heterotrophic. Okay, photosynthetic autotrophs means that they can make their own food by the process photosynthesis, but heterotrophs they can depend other organisms for food. I told you, because they have no chlorophyll pigment or chloroplast. So presence of a chlorophyll similar to plants like, so that it is called as photosynthetic bacteria. And example cyanobacteria, they are unicellular, colonial or filamentous, and freshwater, marine or terrestrial algae. So some of the algae, cyanobacteria, that we can say. Okay, so cyanobacteria is an example for autotrophic type of nutrition of eubacteria, autotrophic bacteria. So cyanobacteria, they are the unicellular, colonial means a group of organisms are living together, more number of organisms, that is called as cyanobacteria. Okay, so colonial, colony. Okay, filamentous, it looks like film. You can see this picture, right side green color. Okay, and it's in a freshwater, marine water or terrestrial algae. So next is the chemosynthetic. Next is the chemosynthetic. So next, chemosynthetic autotrophs. Okay, there are two items: autotrophic, photosynthetic and chemosynthetic, not heterotrophic. Okay, so photosynthetic and chemosynthetic. I told photosynthetic is using sunlight and like a chlorophyll present, using sunlight producing the food, and chemosynthetic using a chemicals to produce the food. Let us see now. They can fix atmospheric nitrogen in specialized cells called heterocysts. So nitrogen fixation takes place here in this organisms. Okay, heterocyst we can say. So oxidized nitrites and ammonia. This is what we can say. Chemosynthetic autotrophs. Okay, chemosynthetic autotrophs. They are also autotrophs. Make their own food. How do they make their own food? They can fix atmospheric nitrogen in specialized cells called heterocysts, and oxidize nitrates, nitrites and ammonia. So heterotrophic bacteria, we will see the third one is the heterotrophic bacteria. Okay, so autotrophic bacteria, photosynthetic, chemosynthetic, heterotrophic bacteria. Now we will see. Majority are decomposers. So decomposer we know very well. It's a microorganism. It's feed on the dead organic matter, dead matter we can say, dead matter, dead organism, dead leaves, plants, pots or organisms, whatever may be. It feeds always the dead organism, that is decomposers. It is decomposing the organisms. So it's helpful in making a curd and fix nitrogen in a legume plants or there you can see the root nodules and all. So the nitrogen fixation for legume plants, it is happening only by means of the bacteria. That bacteria called as that heterotrophic bacteria. So used for production of antibiotics. These are all the useful bacteria. It is giving a use or benefits to the human. Okay. So you can see the first one, nitrogen. It is helpful in making a curd, for curd preparation for all the milk products like yogurt, cheese. Okay, everything is because of the bacteria presence in that, and nitrogen fixation in a legumes plant, and then the production of antibodies in the medicinal field also it is used much. So streptomycin is used to form a streptomycin. Streptomycin is an antibiotic. So production of antibiotic, the bacteria is playing major role in the production of antibiotic. So streptomyces is the bacterial name and heterotrophic bacteria, and it is used to form or produce streptomycin. It is an antibiotic medicine. And pathogens also there. Okay, in heterotrophic bacteria, some of the bacteria are useful bacteria. The uses are the making curd, nitrogen and legume, production of antibodies. Some bacteria are the harmful bacteria, so we can call it as a pathogen. Pathogen means disease causing microorganisms. So pathogens cause disease like typhoid fever, which is caused by means of Salmonella typhi, the bacteria name. It gives harmful effect to the body. Tetanus is the fever, it's a disease, and it is spread by means of a bacteria called as a Clostridium tetani. And citrus canker is a disease, and a Xanthomonas and citri is the bacteria name. Cholera, Vibrio cholerae. So these are the bacteria or the harmful bacteria for Salmonella typhi, Clostridium tetani, Xanthomonas citri, and Vibrio cholerae. So these bacteria can be classified two groups, useful bacteria and harmful bacteria. So useful also we have seen here, harmful also we have seen here. Reproduction in bacteria. Let's see, the reproduction happens both the sexual and asexual reproduction. Spore formation, okay, there are two groups, one is sexual and another one is asexual. First let's see about the binary fission. Binary means division of asexual reproduction, okay, division of one cell into two cells. You can see this picture very clearly. Cell wall is there, DNA material is present, plasma membrane. The cell is elongated and DNA is replicated. You can see the DNA is replicated into two groups in the next picture, and the cell wall is now divided, and finally you can say two separate cells are formed from parent cell. One is only one cell which is divided into same two group of cells. So this is 2 again multiply into divided into 4, 4 divided into 8. Like that the reproduction takes place. This was asexual reproduction or asexual reproduction. Next you can say spore formation. It's a sexual reproduction during unfavorable conditions. Not a favorable, unfavorable condition. You can see this picture here. Vegetative cell is here. And here you can see it is dividing and spore is formed inside. So developing the spore coat next and a mother cell. This is the mother cell, you can see spore coat is formed, well, the red color. Then the spore is coming outside the endospore. Again that endospore is forming a new cell, okay. So we can see that the sexual type of reproduction. So you can see here very clearly in this last picture. So reproduction in bacteria also happened by means of two ways. One is sexual and another one is asexual. So mycoplasma, first we can see, I had told you very first about that cell wall type, right? Two different types, cyanobacteria and mycoplasma. First let's see about the mycoplasma. You can see this picture very clearly. Okay, so mycoplasma is the smallest living organisms without cell wall. Mycoplasma is the smallest living organism without cell wall. Okay, don't forget. So we have seen the cell wall is present in previous bacteria, but in mycoplasma cell wall without cell wall, and most of them are pathogenic. So most of this mycoplasma are the pathogenic organism. It means that it spreads the diseases. PPLO, it means that example it is given pleuropneumonia like organisms. P means pleuro, again P pneumonia, P is solvents, L is a lichen organism, PPLO, and can survive without oxygen. Okay, so the bacteria can be classified into two groups according to the using of oxygen. One is aerobic and another one is anaerobic. Okay, so aerobic can live only in the presence of oxygen, and an anaerobic group of bacteria can survive in the absence of oxygen. So one more is the facultative anaerobic. It means that bacteria can survive both in presence of oxygen and in absence of oxygen. So this mycoplasma group of bacteria can survive without oxygen. So it is able to live even in without oxygen. So now let us see eukaryotic cellular. Okay, so membrane bound organelles are present. We are going to see now about the prokaryotes finished, and now eukaryotic unicellular organisms we are going to see. So the two groups we have seen in the Monerian kingdom, the first one is a prokaryote, second one is a eukaryote. Prokaryote, the organisms, they are made up of unicellular, both the unicellular prokaryote and eukaryote. But the absence of nucleus in prokaryote, U means true, karyo means nucleus, eukaryotes means that the true nucleus present. Let us see about that eukaryote, unicellular organism. So membrane bound organelles are present. Okay, so cell organelles are present. Just a membrane bound cell membrane is clearly visible. Some possesses flagella or cilia for locomotion. So flagella, cilia are the whip-like structure or hair-like appearances around the organisms, which is generally used for locomotion, moving from one place to another place. So they reproduce both sexually and asexually. Okay, reproduction takes place both sexually and asexually. So protista, we can see the protista can be classified into three groups. The first one is protozoan protistans, and next one is saprophytic protista and photosynthetic autotrophs. Okay, these protista can be classified into three groups: photosynthetic autotrophs, protozoan protists, and saprophyte protists. Photosynthetic autotrophs again can be classified into four groups. The first one is, three groups, right? Three groups. The first group is chrysophytes. Okay, the first group is the chrysophytes, and the second one is the dinoflagellates, and the last third one is the euglenoids. So photosynthetic autotrophs can be classified into three groups. The first one is the chrysophytes, and next one is the dinoflagellates, and third one is the euglenoids. Protozoan protists can be classified into four groups. The first one is amoeboid protozoans, amoeba, and flagellated protozoans, the second one. Third one, ciliated protozoans, and the last one is sporozoans. Saprophytic protist is only one type, slime molds we are going to see here. Slime molds that we are going to see. See this table flowchart very clearly. Protista can be classified into three: photosynthetic autotrophs, protozoan protista, and saprophytic protist. Photosynthetic autotrophs are classified into three groups: chrysophytes, dinoflagellates, euglenoids, and protozoan protists classified into four groups: amoeboid protozoans, flagellated protozoans, ciliated protozoans, and sporozoans. Saprophyte protist is only one type, that is called slime molds. So the first one, chrysophytes. You can see these pictures very clearly about the chrysophytes. Some of the examples are given for you. And chrysophytes are the unicellular organisms, and we are studying about the monoderms, right? The unicellular, and they are eukaryotic organisms. As I told you, the meaning for the word the eukaryote, they have the true nucleus. Don't forget. And they are the microscopic organisms, so we cannot see only through the microscope. We can't see these unicellular organisms with our naked eye. They found in freshwater as well as marine water. Okay, so they found in both fresh and marine water, ocean, and it includes diatoms and golden algae. So these are the two groups. It includes diatoms and golden algae. They lack any locomotory organ. So I have told you about the locomotory organ in the previous slides about cilia, flagellum, or something present, a hair-like appearance for the process locomotion, moving from a place to place. But when you see the chrysophytes, they have no locomotory organ. See this picture, you can see very clearly. There is no any whip-like structure or hair-like appearance such as cilia or flagellum. So they lack any locomotory organ and are mostly photosynthetic organisms. So we are studying about the photosynthetic classification only. Photosynthetic means it contains the chlorophyll pigment for the process of the photosynthesis. Cell wall is composed of silica. Cell wall is composed of silica. Don't forget, in earlier we have seen in the mycoplasma, cell wall is made up of the polysaccharides and other things, and a peptidoglycan we have seen in the previous one. And now when we see the chrysophytes, the cell wall is made up of, or the cell wall is composed by silica. And diatomaceous earth is formed by accumulation of diatoms. Okay, so diatomaceous earth means completely diatoms were present place somewhere in the ocean and all. You can see, we can call the accumulation of diatoms, a lot of diatoms, the more huge number of diatoms are together and are forming diatomaceous earth that we can call. Okay, the next one we can see dinoflagellates. Okay, so three groups we have seen. The first one is that chrysophytes we have seen, and dinoflagellates. The next one you can see this picture. Dinoflagellates are always making the red tide in the ocean, and it is giving a harmful effect in the ocean. It absorbs more oxygen, and oxygen deficiency takes place wherever the dinoflagellates present in the ocean, and it looks like a red color in the ocean. We can call it as a red tide. So these dinoflagellates also unicellular organisms, and they are autotrophs. They have that chlorophyll pigment. They are doing the photosynthesis, and they appear yellow, green, brown, blue, or red in color. So different colors of a dinoflagellate we can see. The red color forming the red tide, and yellow, green, brown, blue, or red in color we can see. And a biflagellates, flagellum is present. Flagellum, cilia, I told you, hair-like appearances. So here flagella is present for the movement or for the process of locomotion. So mostly marine organisms, these dinoflagellates found occur only in the marine environment, ocean especially. So you can see here the cell wall is made up of cellulose. Okay, so these are the dinoflagellates characters. Once again I repeat, unicellular autotrophs, they appear yellow, green, brown, blue, or red in color. And biflagellates, they are the biflagellates, and a mostly marine organism, and cell wall is made up of cellulose. So you can see here the red tide. I just have told you can so see through this affected of red tide in the ocean in some of the world, part of the world. You can see United States of America, the location of the Florida. So you can read this data clearly. So next we have to see the last one is the euglenoids. Okay, third part is the euglenoids. So euglena, you can see the flagellum is present, the lengthy part, the hair-like appearance, the topmost region you can see flagellum with this whip-like structure. It is using this whip-like structure, flagellum, for movement. Okay, it's beating in the water and moving a place to place. So they are photosynthetic products. So green color it looks like. So it contains the chloroplast and a chlorophyll pigment, and photosynthetic protists. It is called as the photosynthetic protista, and it is found in the fresh water. They do not have cell wall. So outermost layers made up of pellicle. So this is the important thing. And earlier previous two classes we have seen the cell wall is present. Cell wall is made up of the sign of this one. We have seen one silica cell wall, and the next one we have seen the previous one. Cell wall is made up of cellulose, and here you can see the euglenoids. They do not have a cell wall. Outermost layer is made up of pellicle. Another chemical name is the material name pellicle. It is a highly flexible and flexible layer and made up of proteins. So the outermost layer is made up of proteins and process plant-like presence of chloroplast. Okay, so it possesses plant-like chloroplast. Plants contain chloroplast, right? Like that, this euglena also contain chloroplast. So that we can say autotrophic mode of nutrition. So animal-like character, you can see absence of cell wall. So there are two characters found here. Cell wall is generally absent in animal cell. We know very well clearly. So plant contains the chloroplast. So euglena has both the plant character and animal character. The first one, it contains the chlorophyll green pigment. So it is a plant-like character, and it has no cell wall. So we can call it as it is animal-like character. So that it is a heterotrophic mode of nutrition. So both type of a nutrition takes place here, autotrophs and heterotrophs. So slime molds, the third one is the slime molds. We can see slime molds, you can see this one very clearly. The example picture is given. Saprophytes, they are the saprophytes. So they are the decomposers. It means that it decomposes the dead matter and decay matter, so called saprophytic organisms. So they grow on dead and decomposing matter. So slime molds grow on, you can see in the bread and all that molds are formed after the getting fungus. We can say the fungus. It is so grow on dead and decomposing matter, and found in the dark and damp places. Okay, it's found generally in the dark places, damp places, moisture condition area. And these are uninucleate. It means that only one nucleus is present, so called a uninucleate. So under suitable conditions, they form plasmodium. So plasmodium is also formed. Plasmodium is the protozoans, we know very well. It is responsible for the causing of the malarial diseases. So you can see here this picture, plasmodium, a slime mold, life cycle of this one. See, so feeding plasmodium here, first you can see next mature plasmodium, and the plasmodium matures and sporangia form, that is a spore is forming here, you can see very clearly. Then the spores are here, then flagellated forms, then the organism forms. So have a look on this life cycle of this slime mold. Next to protozoans. You can see the example picture, protozoans. Amoeba is given, Trypanosoma brucei. It's a sleeping sickness, causing sleeping sickness. The worm is called as Trypanosoma. The first one is an amoeba, is the best example for the protozoa. So amoeboid protozoans called amoeba, and the freshwater and marine water. They found both in freshwater and marine water. And it uses pseudopodia for locomotion and to capturing the food. So the elongated part of this amoeba we can call it as a pseudopodia. Pseudopodia, podia means leg, pseudo means false legs. It is used as a leg, but it's not a leg, okay, so that it is called as pseudopodia. This pseudopodia is mainly used for the purpose of locomotion. And holozoic mode of nutrition takes place here. Holozoic means it's engulf or phagocytic. We have seen the phagocytosis, right? Engulfing the food substances, that is called as the holozoic mode of nutrition. And a flagellated protozoans also present. And free-living parasites, they are free-living. It means that it is not touching any substrate, and instead it can swim in the water area or moving here and there everywhere, freely moving. So that free-living or parasitic. It possesses flagella. And the trypanosoma, you can see it causes a sleeping sickness in a human. So Trypanosoma brucei, the picture is given here. You can see clearly this picture. Trypanosoma causes a sleeping sickness in humans. Next, ciliated protozoans. Let us see now. You can see this is a paramecium is an example for this. The body is completely covered by outer side, the hair-like appearance you can see. This is called as a cilia. This is cilia is used for the locomotion, moving from a place to place. So they are aquatic organism. It is living in the water area only, and locomotion with the help of a cilia. And next is the sporozoans. Example, you can see the Plasmodium vivax. So the Plasmodium vivax is an example for malarial parasite, and the sporozoan have infectious spores like a stage called Plasmodium. It's a stage of the spores and sporozoans, and it is a pathogen. Okay, it is a pathogen, and it causes the infectious diseases such as malaria. Example, Plasmodium. So next, let's see about the kingdom. Monera finished, and next we have to see about the kingdom fungi. So kingdom fungi. First, you can see here fungus. We know very well, yeast, mushroom, all are example for fungi, right? Penicillium is also the fungi we can say, notatum, Penicillium notatum. So this fungi is generally classified on the basis of the structure of the cell, body organization, then presence of the cell wall. Okay, yeast is the best example. Antibiotics produced from this fungi, and wheat rust is also another example for this fungi. So let's see the body conditions, body structure. The body is made up of hyphae. Hyphae is a hair-like appearances. So we can see this picture very clearly, the hyphae. Okay, so body is completely made up of the hyphae. You can see this picture clearly. Okay, so network of hyphae is called mycelium. Many hyphae are joined together. Together we can call it as mycelium. So the body is made up of hyphae. The network of hyphae is called as mycelium. And nutrition can be saprophytic. Saprophytic means that it feed on the dead organism. Fungus, we are using the word. If spoiled to food items, we are saying the fungus, right? So they are called as the saprophytic. It feed on the dead organisms or decay matter. And next to parasitic, attaching with the other organisms with the host, giving a harmful effect to the organism. And symbiotic relationship and attaching with other organisms and giving benefits from each organism, both organisms. That is called as the symbiotic. So three groups we have to see here: saprophytic, parasitic, and symbiotic. So next we have to see about the kingdom fungi can be classified into two major groups. The first one is aseptae and the second one is septae. Septae means septum. Septum means separation. So dividing, that is aseptae, absence of separating or absence of septum. Presence of septum is or separating is septae, we can say. So the aseptae fungi can be classified into as phycomycetes, and septae can be classified into three groups. The first one is called as ascomycetes, and the second one is called as basidiomycetes, and the third one is called as deuteromycetes. So fungi kingdom can be classified into two groups, aseptae and septae. Aseptae is classified into only one type, that is a phycomycetes, but septae can be classified into three groups. The first one is ascomycetes, second one is basidiomycetes, and third one is deuteromycetes. And first let us see about the aseptae, phycomycetes. So you can see here the picture clearly, example is given. So it is the aseptae and cenocytic hyphae. Okay, what is meant by cenocytic hyphae? So it is found in aquatic habitat and on decaying wood in moist and damp places. Damp means that wet conditioned area also we can say. Okay, so it is found in habitat and on decaying wood in moist and damp places. Asexual reproduction by zoobos and aplanospores. So sexual and asexual both happens here in this kingdom, and asexual reproduction by zoospores and aplanospores. These are the two responsible for this asexual reproduction, and sexual reproduction takes place by the formation of zygospore. Example, rhizobus, bread mold. So it is male forming in the bread mold. It's called rhizopus. So both type of reproduction found in fungi, in phycomycetes, that is aseptae. One is sexual and another one is asexual. Asexual reproductions found by means of zoospores and aplanospores, and sexual reproductions found by means of zygospores. Example, rhizobus, it's a bread mold. And next we can see here about that reproduction. How does it takes place? Zygospore, it is given here, sexual reproduction by zygospores. So organisms and a small blood rises as a nuclei is transferred. Here you can see the nuclei and zygoplasm, and from there the zygospore is detached separately, and the zygospore is forming a meiosis. It undergoes meiosis to produce a form of spores. Lot of spores here germinate here, males and females, everything. So this is a strains develop into new hyphae. Okay, so this picture give you clear information about that zygospore, sexual reproduction by zygospores. Can see this picture clearly. So next is the ascomycetes. Okay, the first one, phycomycetes, one is aseptae. So septae, three types I told you: ascomycetes, basidiomycetes, and deuteromycetes. In which first we have to see about ascomycetes. Ascomycetes, you can see this picture clearly, And mycelium, mycetes, it's always in the word the mycelium. So mycelium is a branched and septate. Septate means separated. You can see here picture clearly. The mycelium is that hyphae is separated, so that we can say this is a septate group. So asexual reproduction, the asexual spores are conidia produced. Here the asexual reproduction found, and asexual reproduction is formed by means of the conidia, and exogenously on the special mycelium called conidiophores. Conidia on germination produce mycelium. So exogenously the special mycelium called conidiophores. So the conidium is present, that mycelium is called as the conidiophores, and conidia on germination they produce the new mycelium, new group of organism, a mycetes. Okay, this is called as ascomycetes. And next is the basidiomycetes. Okay, before that we can see the sexual reproduction. How does it find? You can see this picture clearly. So which are produced endogenously in sac-like assay, that is singular ascus we can see. See this picture clearly, it is given asci is formed, and sexual spores are called ascospores. And these assay are arranged in different types of fruiting bodies called ascocarps. See this picture, then you can understand clearly. So next one is the basidiomyces. Okay, basidiomyces, and see this is a mushroom is given an example is the best one. Caps are there. So grow in soil on logs and tree stumps. So mushroom generally we know during the rainy time you can see when the climate gets the moisture, it grows in the wooden pot and the logs, in the soil, tree, we can see everywhere, right? The mycelium is branched and septate. So mycelium is branched and septate, this is we can call. So asexual spores are generally not found here, and sexual reproduction happens here. Plasmogamy and dikaryon and karyogamy. Okay, and basidiospores. So you can see the sexual reproduction found in the basidiomycete. Plasmogamy, it is a union of cytoplasm, and dikaryon, it means that two nucleus present, and karyogamy, union of nucleus, occur in basidium, and basidiospores are also present. See this picture clearly, sexual reproduction by basidiospores. Okay, you can see the basidiospores and primary mycelium, and from which the spore forms, then this one is formed finally. Okay, so taxoma, see this picture, you can make pause and see this picture clearly, everything is explained. Sexual reproduction found in basidiomyces. And the last one is the deuteromycetes in septate, third class. So this class of fungi is known as imperfect fungi. Okay, so it's not a perfect fungi, it is, we can call this the imperfect fungi. So because only vegetative or asexual phases of these fungi are known, so that we can call it as imperfect fungi. Once these sexual stages of fungus are identified, they are transferred to ascomycetes or basidiomycetes. Okay, so once the sexual stages of fungus are identified in deuteromycetes, they are transferred to other two parts, ascomycetes and basidiomycetes. So trichoderma, colitotrichum and alternaria, these are the best example for this deuteromycetes. See this table, different types given here very clearly: Phycomycetes, Ascomycetes, Basidiomycetes, Deuteromycetes. So it is algal fungi reproduce from spores. First, Phycomycetes, an example, Mucorrhizopus. And Ascomycetes is a cyclic fungi reproduced from ascospores. An example, Aspergillus and Penicillium is the best example for Ascomycetes. Basidiomycetes, it is a glub fungi and reproduction takes place by fragmentation. An example for this is as agaricus and mushrooms. Deuteromycetes is imperfect fungi, sexual reproduction found here. Once it happens, for what happened? It is transferred to Ascomycetes and Basidiomycetes, I told you. Example, Alternaria and a Trichophyton. So these are the different types and characters you can see. So next we have to say about the virus. We know very well about the virus, and all over the world now is affected by the coronavirus, and we all have no very clear-cut information about what is virus, right? So the name virus means venom. Venom means what? Poison or poisonous fluid, and it was given by Pasteur DJ Ivanovsky. He is the scientist who first explained about this virus contains venom fluid, and it is given by Pasteur DJ Ivanovsky. He studied the cause of disease, tobacco plant. First, he actually discovered from this in tobacco plant. Okay, so he studied the cause of disease, tobacco plant. He showed that the disease of tobacco was due to virus. It's called as tobacco mosaic virus. First, he invented this virus from the tobacco disease. He actually undergone a disease research for the tobacco diseases, plant disease, and he found that there was a virus which is responsible for causing the disease in the tobacco plant. So he gave the name that is called as tobacco mosaic virus, TMV. And the next, M.W. Bijurny in the year 1898 demonstrated that infected tobacco plant could cause infection in the healthy plants. He called this fluid Contagium vivum fluidum, okay, infectious living fluid. If one plant is infected, it completely spread to all other plants in the field. That's what demonstrated by that Bijurny scientist, okay. So infected tobacco plant could cause infection in the healthy plants as well. He called this fluid, he actually called it as a fluid that is called as Contagium vivum fluidum. So these are the scientists, two scientists you can see, the Bijurny and Pasteur Ivanovsky. So next you can see Stanley, another scientist, W.M. Stanley, showed that viruses could be crystallized, and crystals consist largely of proteins. And he explained clearly about the structure of the viruses. Viruses could be crystallized and crystals, and it consists large of proteins. It contains proteins inside it. That's what explained clearly about the Stanley, the last scientist. So, can see here the important points about this. Viruses are the smallest infectious agents and contain either Na or DNA, but not both together. Okay, nucleic acid or deoxyribonucleic acid. Okay, so viruses are the smallest infectious agents and contain either Na or DNA, but not both together. They are obligate parasites. Parasites can live only in the host organisms, and it cannot survive out of the body of an organism. It's a non-living thing when it is out of the body. When it reaches the host organism, living organism, it becomes a living organism, starts multiplication. And they are insert outside their specific host cell. Okay, so they are inert outside. Inert means inactive outside the host cells, outside the organisms. They are able to pass through the bacterial filters. Viruses do not possess all the enzymes necessary for multiplication. Therefore, they are dependent on the host cell. Viruses must not contain all the enzymes required for the reproduction. That's what it is, getting everything from the host cell, so that parasites we can say. See the picture of the virus. I think everybody knows the structure of the virus nowadays clearly, and we came to know more information about the virus because of this corona disease spread, pandemic disease now, right? So structure of bacteriophage. What is meant by bacteriophage? You can see this structure of virus here. We can say the bacteriophage. What is meant by bacteriophage? These are the viruses which infect bacteria. So some of the virus affecting the bacteria called as the bacteriophage. Okay. So it has our sheath. It has outer sheath of protein coat enclosing a DNA molecule. So DNA molecule is present inside and it is covered or enclosed by a outer sheath or covering. It's made up of protein. And the head, there are parts, you can see the head and the tail. There are two parts given here. Head, protective covering for the nucleic acid, double-stranded DNA is present. And tail, hollow tube through which the nucleic acid passes during infection. So through this tail, the nucleic acid passes to the host organism. Okay. So it is surrounded by base plate and tail fibers. So a lot of tail fibers you can see in the bottom side, and the basic plate is there. From the basic plate, a lot of tail fibers are rising outside. Tobacco mosaic virus. Structure of tobacco mosaic virus. TMV means that tobacco mosaic virus is a long, cylindrical and rod shape. You can see the picture, and it is made up of a nucleoprotein, protein and nucleic acid, so called nucleoprotein. And central core is made up of RNA. Central part is made up of RNA. And external sheath is made up of a protein coat. It is called capsid, is made up of identical subunits and called, you can see, external sheath is made up of a protein coat. It is called capsid, and capsid is made up of identical subunits called capsomeres. Okay, the sheath is outer covering is called as the capsid. It contains capsomeres inside it. So viroids, okay, viroids, what is meant by viroids? Viroids discovered by T.O. Diener in the year 1971, and it comprises of small single-stranded molecule of RNA, and it is smaller than viruses. Viroids are smaller than viruses. Lack of protein coat found in viruses and have no proteins associated with them. Next one is the prions. So virus finish, viroids, and next is the prions. So it is the infectious agents with misfolded protein structures, and these are smaller than viruses, and it causes brain diseases in mammals. So prions are the one mainly causing the brain diseases in mammals, even in human. So bovine spongiform encephalopathy, commonly called mad cow disease in cattle, and its analogous variant, see your Jacob disease, CJD, in humans. So these are the diseases are caused by these prions, okay, BSE and CJD. Let's see detail. You can see here how the crudes filled to Jacob disease oaks, the brain disease of a human. Okay, it's an example given in this slide. You can just pause it and go through this slide. You can understand the cause and consequence of these diseases, the pyroids diseases. Okay, how these pyroids diseases are caused. So lichens, last one is the lichen. So it is a symbiotic association between algae and fungi. So it is a symbiotic relationship between algae and fungi. So lichens are actually it is in the fungi and algae, and it gives what we can say it both organisms are benefited. That is called a symbiotic relationship. So phycobiont is an algae, and mycobiont is the fungal component. So these two are living together and getting benefit from each organism, getting oxygen from this, and algal, that fungi is getting oxygen from there and giving food for that organism. So both organisms are getting mutual benefit from each organism. phycobiont and a mycobiont, algal and a fungal component. So this is called as the lichens. Lichens are a good pollution indicator. See this picture very clearly. Fungal hyphae is present and algae is present inside. Okay, thank you students. I hope everybody understand very clearly about this lesson, and I give you a suggestion if you have any doubts and any modifications, you please give your suggestion in the comment box. And then if you don't subscribe, please subscribe to the channel for getting more lessons. I plan to upload all the lessons in grade 11 and 12 in biology. And if you like this video, please give your likes. Okay, click the like button and share to your friends. Okay, thank you very much for watching this video.